Welcome back to the Biology 1440 Lab. Today I'm doing a video showing you how the photosynthesis experiment would be set up if we were meeting in person. So what do we have going on here today? We have a light, a white light, you can see here, quite bright. It's an LED light. We have in this case, a red filter. And down here we have a leaf inside of this container. I laid it in there as flat as I could so it would be exposed to the light. If we were doing this in lab, we'd use spinach, but I grabbed an oak leaf from outside. And we have a carbon dioxide probe right there. It reads concentration of carbon dioxide in the atmosphere. Okay, so right now I am incubating this plant. It is sitting in there in this reddish light. And what we're going to do in a little bit is we're going to collect data on this computer, and we're going to see how much CO2 concentration changes over a 5-minute period. So you would want to make some hypotheses. First of all, what should carbon dioxide levels do if the plant is photosynthesizing? Something to think about. And what we would do in class then is we would try white light, red light, blue light, green light, and darkness to see what photosynthesis does in each of those conditions. More importantly, we'll see what carbon dioxide levels do in each of those conditions. So your job would be to write a hypothesis or several hypotheses that takes into account what the plant would do with carbon dioxide at red, white, blue, green, and no light. Okay, I think I got all of them listed there. And we would run this and replicate everything so we can get enough data to make a figure with error bars and things. So I'll come back in just a second. Okay, we've been incubating for about 5 minutes, so I am now going to take the probe, the CO2 probe, place it into the chamber, and I'm going to click what looks like the play button here, which is collect data. Now what it's going to do, it's going to read carbon dioxide levels in parts per million, and it's going to start making a graph, and it's going to run for 5 minutes. And so what we will do is get the slope of that line, at least of the last few minutes of that line after it runs. So I'll come back in a few minutes. Okay everybody, here's the data we get after a 5-minute period. So I'm going to get the slope of the last half of the data here. So I'm going to highlight the data, and I'm going to click the linear fit button, and we can see that the slope is negative 12.21 parts per million per minute. So remember, a slope is a unit over time, just like miles per hour. In this case, it's a negative number. What does that tell us? A negative value tells us that over each minute there was about 12 parts per million of CO2 absorbed every minute by that leaf. It was pulling CO2 out of the air. Now what will happen if we put this leaf in darkness and we run this experiment again? What is your prediction? Okay, we're back in lab a few minutes later here. We've had the leaf sitting in essentially darkness. I didn't wrap it in foil, but this room is pretty dark to that leaf. It's not able to photosynthesize with just those lights up above. And I let this data run for 5 minutes, and we can look at what happened. And now we have a slope that is 4.353 parts per million per minute. So we have a positive slope. That means that this leaf was releasing carbon dioxide. So if put in darkness, it will respire. It always is respiring, but when it's photosynthesizing, it's absorbing more carbon dioxide than it's releasing. But in darkness, it cannot photosynthesize, and it respires just like you do using its mitochondria, and it releases carbon dioxide into this respiration chamber. So what we would do then is we would change out filters. We would try different colors of light, and we would do replications. And then we would take that data and graph it and see if our hypotheses are supported or not. So that's what we'll probably do for an assignment. Stay tuned for another video or check out the handout on what the assignment actually is regarding that data.